PG for peptidoglycan. Down here, this is the cell membrane. Okay. The lipid bilayer, we'll talk more about that. And then this, these long spikes there that's kind of anchor the peptidoglycan down into that lipid bilayer. That's the lipoteichoic acid. And then the little spikes that don't extend into the plasma membrane, that's the teichoic acid. Right. Okay, so again, not a whole lot of parts there. In terms of the shape of the cell, sometimes students have the mistaken notion that, oh, all gram positives are one shape and all gram negatives are another shape. That doesn't necessarily have anything to do with it. So there are gram positive cells that are cocci, that are rods, that are vibrios, and there are gram negatives that are all those things as well. So, okay, let's look more closely at the gram negative bacterial cell walls. Okay, as you might expect, they have a thin layer of peptidoglycan. Yeah, only about 10% of the dry weight of the cell wall is peptidoglycan. So it does have some, just not a lot. The really unique thing is that the gram negatives have an outer membrane. So in addition to their plasma membrane, and then their peptidoglycan layer, on the outside of that, they have a whole nother lipid bilayer, similar to their plasma membrane, but it has a few other features to it as well. It has some proteins in there, some special protein channels called porins. Okay, so those are protein channels that are in the outer membrane. Okay, and we don't see that in gram positives because gram positives don't have an outer membrane. And also a compound called lipopolysaccharide. Okay, we usually abbreviate that LPS. Okay, and that's composed of two different polysaccharides that we really don't talk about too much. I'll show you on the diagram, as well as lipid A. And lipid A has another name that's a little bit confusing, also known as endotoxin. Okay, so just keep that in mind. We will talk more shortly about endotoxin. All right, so if we label these things on your book's diagram, this plasma membrane, of course, is on the inside. This pink kind of Wendy's hamburger type structure here, that's peptidoglycan. Okay, only kind of a very small amount of it. And then outside of that, that's the outer membrane. Okay, so over here, these two kind of looks like maybe paper towels together here. That's a protein channel called a porin. Okay, for transfer it through that outer membrane. And then these kind of hair-like structures out here, that's the LPS, the lipopolysaccharide. Okay. Right, and that's just, I stole the figure from your book and took all the labels off of it, so you can find it there in your book with the labels on. Okay, so I do like this picture a little bit better. This is from another Pearson textbook. I like this figure a little bit better of the gram-negative cell wall because it shows a little bit more detail parts of the lipopolysaccharide. Okay, so those hair-like structures, those are the two polysaccharides, sugars kind of sticking out there. And then you can see really a little bit better how that lipid A is really embedded right down in the membrane there. Okay, so the significance of lipid A, I said that it was also referred to as endotoxin. And that seems like a really strange name because it seems like endo, it should be inside of the cell. Well, we originally thought that it did come from inside the cell, and then we realized later that it was actually on the outside of the cell. But the name stuck. We call it endotoxin, and this is significant for gram-negatives that are causing disease in our body. Okay, so if we have a gram-negative infection, that lipid A is released anytime a cell dies. Okay, anytime a cell dies, the cell wall starts to break apart. Okay, so when the cell wall breaks apart, then that's going to release the lipid A. Okay, so it's released when the cell dies. Okay, so what's the so what of that? Well, like you might guess from the name endotoxin, that causes some harm in our body. Causes some increased inflammation. We're going to talk much more about inflammation later on in the course. That's going to cause things like fever, blood vessels get a little leakier, some more clotting takes place, kind of non-specific things, usually kind of low-grade effects. But if you have a gram-negative infection and a bunch of those gram-negative cells die all at once, and you get lots of endotoxin in your system, it can cause the patient to go into shock, or blood pressure to go way down. So for that reason, we need to kind of know what kind of infection somebody has, because if it's gram-negative, we might be better off not treating it and let it run its course. Or we may treat it and then add lots of anti-inflammatory drugs as well, so the patient doesn't have lots of inflammation. Okay. All right, but that again, lipid A is endotoxin. It's the same thing. Okay, so that's gram-positive versus gram-negative structure. There is actually one other group, a different kind of cell walls that is only actually a couple genera of bacteria that have this kind of specialized cell wall, and it's called acid-fast. So these are bacteria that really don't take up that gram stain very well at all. So we don't consider them gram-positive or gram-negative. They're in their own special group. And this is the genus Mycobacterium. And we have to have a special staining procedure to see these cells. We call it the acid-fast staining procedure. And it seems like a lot of trouble to go to just for, kind of only a few bacteria. But this genera causes some pretty significant infections. One of them, well, it's not so significant anymore, but you've probably heard of leprosy. And the other one is tuberculosis, still a major scourge worldwide. So again, we had to have a special staining procedure because what's unique about these cell walls is, so the cell walls have a high concentration of mycolic acid. So these are cell walls that have peptidoglycan in them as well, but they also have a lot of other stuff. One of the things is mycolic acid. This is a long chain fatty acid that basically gives the cell wall kind of a waxy feel, or we could say appearance, I guess. I might not have spelled that right. Okay, so waxy appearance. So basically, because there's all these fatty acids in the cell wall, when we try to gram stain these cells, the gram stain dyes are dissolved in water, And, you know, if you put water on wax paper, it just kind of beads up, right? It doesn't penetrate in there. So we had to have a whole new staining procedure. And that's where the name acid fast came from. When we decolorize I won't go through the staining procedure, you're going to do it in lab if you're in lab. But when we try to decolorize these cells, we actually use a really potent decolorizer, hydrochloric acid dissolved in alcohol. And if organisms still hold on to the primary dye, they hold fast to that primary dye, even when decolorized with acid. So that's where the name acid fast comes from. Okay, most cells, gram positives and gram negatives, will be decolorized, and then they'll take up the counter stain. So in this stain here, the non-acid fast ones are blue, and the acid fast ones are hot pink. They hold on to that primary dye. Okay, but again, I'll leave that for your lab. It's just kind of, that's where the name acid fast came from. Okay. All right, so I found this slide that kind of compares all three of the different kinds of cell walls that we find in bacteria. There are actually bacteria that don't make cell walls. There's a genera that they're called mycoplasma that don't have cell walls, but most of them do. So remember the gram positives, you can not worry about this. Lots of peptidoglycan, gram negatives, outer membrane, less peptidoglycan. And the mycobacterium, the acid fast cells, they do have some peptidoglycan. Major thing here on the outside is the mycolic acid. So don't worry about some of this other stuff that's in there. It's a little bit more complicated than that, but we think a lot of its unique properties are due to that mycolic acid. Okay, so little thought question here. So if we're thinking about patients that have infections with, you know, all three of these different kinds of bacterial cells, which one of these cell walls do you think is the hardest to kill? All right, so if we're treating a patient with antibiotics, which one of these kinds do you think might be toughest to kill? What do you think? Ah, good. Okay, yes, in fact, the mycobacterium are definitely the hardest here because they're just not, it's just not permeable, right? I said the gram stain dyes can't penetrate it. We only have a very short list of antibiotics that are able to penetrate through that waxy cell wall. And so definitely things like tuberculosis are very difficult to treat. Long regimens with antibiotics, long periods of time. That mycolic acid gives those acid fast cells pretty good chemical resistance in the environment as well, and certainly against antibiotics as well. The second most hardest one to kill are the gram negatives, because again, they have this lipid bilayer. Remember, it's the membranes that are selectively permeable. So it's harder to get antibiotics that were able to penetrate through that outer membrane. You might have thought gram positives because it's got all this peptidoglycan, but remember we compared that to a chain link fence. And as long as something can fit through those holes, you know, it can go right through. So it's kind of dumb, right? Like, even though it's strong and rigid, like lots of things can just sieve right through it, I guess. So all right, that's just kind of thinking ahead to when we talk about antibiotics a little bit. So good. All right, so moving forward into the cell, I'm not going to spend very much time on the cytoplasmic membrane here. Hopefully this structure is familiar to you. It's very similar to what our cytoplasmic membranes look like. Remember the function, selectively permeable. It's controlling what comes in and out of the cell. It's really important to have intact cell membrane when the cell needs to generate ATP. There's only certain channels where the ions can come in, so we have to only let diffusion basically take place in certain channels. And if this was a photosynthetic organism, then it would have pigments in the cell membrane. Well, yes, okay. And of course, like glycolysis takes place, well, in the cytoplasm, it's the electron transport chain that's in the plasma membrane, okay. Thinking about transport through that membrane, any process that's passive, that means ATP is not required for it to happen. So things like simple diffusion, a substance moving down its concentration gradient from high concentration to low concentration. What kind of things can diffuse through a cell membrane? Really small things like simple sugars. When water diffuses, we call it osmosis. Some gases, anything that is soluble in lipids, like alcohols, can go through. It's kind of a short list, though. Other kinds of transport that don't require energy, facilitated diffusion. So that is facilitated by some kind of protein channel, and that can be non-specific in that kind of anything can come through, or very specific. Oftentimes we refer to that as some kind of permease enzyme. For example, to get lactose into the cell, that doesn't require energy, but there has to be a special enzyme, a permease, that's going to allow it to come in really quickly, and just lactose. And then osmosis. This is the diffusion of water. So let's review that a little bit. It's always confusing for some people. I think maybe where I used to get confused about the whole concept of these terms, isotonic, hypertonic, hypotonic, these are always going to refer to the solution that the cell is in. Okay, it's not correct to say the cell is hypertonic to the medium. Okay, that's not correct, because the reason that we call the solutions those terms is because the solution is likely to change pretty quickly, where the chemistry inside of the cell is not likely to change very quickly. So always use those terms to refer to the solution that the cell is in, and then thinking about which way the water moves in which instance. Of course, in an isotonic solution, there's no net change in water, same amount is leaving as amount what's coming in. In a hypertonic solution, that means there's more solutes outside the cell, solutes that can't penetrate through the membrane. Usually we say like sugar or salt as an example. And because there's more solutes outside, the cell is trying to equilibrate the inside with the outside. So water is going to be leaving the cell. Some people say water follows stuff. Okay, there's more stuff on the outside, the water is going to follow it. Okay, so this is a pretty bad situation for a bacterial cell, or for any cell when it's dehydrating. Okay, it's going to crenate. Here we have the cell membrane kind of pulling in from the cell wall. Okay, most cells cannot survive that for very long. Some organisms have special adaptations to be able to live very comfortably in very hypertonic mediums, but most of them not. That's why we add salt when we're trying to preserve our food, to keep bacteria from growing, or sugar even. Okay, and then in a hypotonic solution, that means low solutes. So if the solutes is low, the water concentration is going to be higher outside of the cell, and it's going to be moving into the cell. You can see when we compare cells with a cell wall with cells that don't have a cell wall, there's a big difference in what happens to that cell. All right, bacteria cells have this nice cell wall that protects it. Okay, as long as the cell wall is intact. If it's growing and there's some drug in the way that's kind of leaving little holes in the cell wall as it's growing, then we can get the bursting like we would in cells that don't have a cell wall, lysing. Okay, so typically bacteria can withstand being in a hypotonic environment much better than, say, a red blood cell that would burst. Okay, what about processes of transport that require energy. Okay, we call that active transport. So I'm not really too worried about this terminology, uniport, antiport, all that. The idea is that, do you know what active transport is? It means that cells spending some energy to bring something into the cell. Usually there's going to be some kind of protein that's necessary for that to happen. And whether, sometimes the cell lets one thing diffuse out, and then when it comes back in again, that makes it more energetically favorable. That's what we see happening over here. But main idea is energy is spent. And then there is one kind of transport that is unique to prokaryotes. Okay, so bacteria do this and we don't. And that means that, and this is called group translocation. It's not an intuitive name to me. I'm not sure why they call it that. But this just means that whatever compound that's being transported in actively is changed in the process of being brought into the cell. So the best example I have of that, this is actually the first step in glycolysis, where glucose is brought into the cell, and in that process, it's changed to glucose 6-phosphate. Okay, so why would it tag that phosphate on there? Well, the cell is trying to concentrate glucose inside of it. All right, remember, glucose is one of those things that can just diffuse right through that membrane. So if there's a lot of glucose inside the cell, some of it is going to be lost. It's going to go from a high concentration to low, unless you modify it a little bit. Okay, now with that phosphate on there, it's changed and it can't just diffuse away again. So that's kind of a strategy for the cell to concentrate, something that it needs. All right, group translocation. All right, so moving into the cell. Like I promised, there's not a whole lot in there, so this will go fairly quickly. Of course, there's cytoplasm in the cell. Technically, cytosol, in the past, I've used the term cytosol and cytoplasm interchangeably. Technically, the cytosol is the fluid part of the cytoplasm. So technically, cytosol is part of the cytoplasm. I'm not too worried about splitting hairs there. What else is in there besides fluid? There's ions, proteins, just a couple of examples. In prokaryotic cells, remember, don't have a membrane-bound nucleus, but they do have a nucleoid region. So all of the chromosome or chromosomes inside the bacterial cell are typically, again, all in one spot, even though they're not, covered in a membrane. Okay, this is where the DNA is located. Okay, we'll talk a little bit more about that prokaryotic DNA when we get to our genetics, but for now, know that they typically have one, sometimes two chromosomes, and they are typically circular. Okay, kind of like what you see down here. It's just, that's a way to protect the ends. There's no ends that are going to get degraded by any kind of nucleases or anything. And they also have DNA in the form of plasmids. So plasmids are just going to be shorter pieces of DNA. Usually that DNA encodes for things that are not absolutely essential for the survival of the cell. Might be something like antibiotic resistance, something that may give it an advantage in certain circumstances. So we'll talk a little bit more about that later, specific kinds of plasmids. Okay. One very important structure inside the bacterial cell is the ribosome. And of course, this is where proteins are synthesized. Okay, so ribosomes actually don't have a membrane around them. Okay, so bacteria have them as well as eukaryotic cells. And it seemed like a reasonable thing just to talk about eukaryotic ribosomes at the same time as prokaryotic ribosomes. They are made up of ribosomal RNA and proteins. And we have a way to measure the size of these pieces of ribosomal RNA. They are made up in two, I'll get to that in a minute, two subunits. So you can see here, these are the prokaryotic ribosomes, and the eukaryotic form is just a little bit different. But we have a small subunit and a large subunit, and those only come together when they are actually making a string of amino acids into proteins. Okay, the units that we use here, so you see this 5S, this 23S. S stands for Svedbergs. Okay, and that is a unit of sedimentation rate. So basically, when you put ribosomes into a centrifuge tube and spin them around, the rate at which they fall to the bottom, that's the sedimentation rate. So it's kind of a measure of the size and the weights and kind of the structure of the ribosome. And that's why it's not linear. So when we say we have a 50S subunit and a 30S subunit, that adds up to 70S. Okay, it's not really linear. And of course, the eukaryotes have the 60S and the 40S that make an 80S ribosome. So kind of the same kind of composition, but a little different size. Even the pieces of ribosomal RNA, like the 5S and the 23S, remember there's proteins added in there as well that make that whole subunit measure 50S. Okay. And because the differences between the prokaryotic and the eukaryotic ribosomes are fairly slight, but it's enough that we can capitalize that. We have a lot of antibiotics that target prokaryotic ribosomes, and they don't have much effect on eukaryotic ones. So again, anytime there's differences there, we can capitalize that. And in case I don't bring up the archaea very often. I say prokaryotic. The archaea actually have ribosomes that are very similar to the prokaryotic ones, the same sizes. They have a little bit different shape to them, so they are a little bit different, but they're most similar to prokaryotic ones. So I just threw them in there. Yes.
I'm sorry, but I'm just confused on how with 50 and 30 cannot.
Right. So that's a great question. Because it's actually a sedimentation rate, it's not something, like I said, it's not linear. So it's not like it's this mass and this mass that would add up. It's more of a unit that takes into account shape and weights and things like that. So that's kind of the best answer I can give you right now. I can look more into that, but it's just a measure of how fast it falls to the bottom when we centrifuge it. So, yep, good thing to be curious about. Okay. All right, so pretty much that's it for a lot of bacterial cells. Some bacterial cells can have endospores. So we talked about this briefly when we were talking about, hey, these are ways that we can look at characteristics of cells to classify them. There are really not very many bacteria that are able to produce endospores. two genera, the Clostridium and Bacillus. I mentioned them because you'll be using both of them in lab if you're in lab, but those are the most famous organisms that are able to sporulate and make endospores. So basically, if you look at that sporulation cycle, if you start with a vegetative cell, and that means a metabolically active cell. Okay, so the cells that say you're growing up on a plate, if that cell encounters some kind of harsh environment, so examples there might be low nutrients or a temperature that's not ideal, or maybe exposure to radiation or pH fluctuations. Anything that is not ideal for the cell, it can cause it to start to sporulate. So you can see how it's, this is showing the DNA being replicated and it being concentrated in part of the cell. Sometimes that happens at the end, sometimes in the center. It depends what kind of cell it is. For instance, I think all the bacillus do it right in the center of the cell. And then a spore coat develops around this. And we could spend a long time on talking about the structure of that. There's many different layers. A protein layer with keratin in it, which is the structural protein in your fingernails. It's kind of dehydrated in there, but a really tough spore coat, and eventually the vegetative cell drops off and you're just left with this free endospore. Okay, and that process takes somewhere between 6 and 10 hours. So it's not going to, if it's, if the environment very quickly changes, it's going to kill the vegetative cell. If it's very slowly changing, then it will have a chance to sporulate. And that free endospore can survive years, decades, thousands of years. We don't really know the upper limit of that. So these are really tough structures, and then they will germinate whenever the environment becomes favorable again. Okay, so we think that maybe some cells are maybe just constantly going through that spore cycle, so that if the environment does change very quickly, at least some cells might have the chance of surviving later on. Okay, so it's, again, I think I said this before, this is a survival mechanism. It's not considered reproductive because you're getting one endospore from one cell. Okay, so it's just kind of hunkering down and surviving. And it takes a lot of unique genes to be able to do that, so it's not common in the bacterial world. If you are in lab, you will get a chance to do a spore stain, which should look something like that. Just show you a picture of that. Bacillus anthracis, anthrax is a spore producer. If you were to just say add a single dye, like is done right here, we have to have a special spore staining procedure to be able to dye that spore coat. And here you can kind of see the free endospores, they don't take up the dye very well. Because so if you gram stain it, even like the spores don't show up very well. That's why we have a special spore staining procedure. But that's cool looking, I've always thought. Another thing that only some bacteria cells may have are inclusions. And these are just storage sites for something that the cell might need more of later. Okay, so typically lipids, starch, some other necessary chemical. And one really interesting example of organisms that can do this, I think Bacillus, the organism that we were just talking about that forms spores, is capable of doing this, but it's capable of making polyhydroxybutyrate inclusions, or PHB. This is just a starchy polymer. Again, just kind of storing it up if you grow it in certain kinds of mediums, kind of storing it up for later. And these are not membrane bound. There is some kind of barrier that keeps that starchy polymer from being released into the rest of the cell, but it's not a membrane And what's really unique about this, we can cause bacteria, we can grow them up in a medium that can cause them to make lots and lots of PHB. And then we can use that, we can extract that PHB and make it into an organic plastic. All right, so I just love this, the idea of using bacteria so to kind of make our life better. So here's a water bottle that's kind of been thrown in a landfill, and you can see if it's made out of PHB, it degrades, you know, over a matter of weeks or months, rather than sort of never with petroleum-based plastics. So that's kind of a good idea. Maybe for some applications, you don't necessarily want your plastic to degrade quickly, but it's kind of a cool concept. Right now, it's a lot more expensive to make plastics with PHB, so we have to kind of get the cost reasonable before that's going to be used widespread. But it's kind of a cool idea. So we'll just write here organic plastics. Okay, cool idea. Another example of a kind of inclusion, there are bacteria that will kind of concentrate gases within their cell, and that's in order to place themselves in the appropriate spot in the water column. Maybe if they are photosynthetic, they need to, you know, kind of get a certain amount of light. Okay, so it helps them, we'll say, for flotation. All right, so pretty much that's it. We could talk about the cytoskeleton a little bit, but in your book denotes a paragraph to that. But I think we will just move on and talk a little bit now, switching gears to eukaryotic structures. Some of the organelles I'm going to skip through really quickly. I'm just kind of basically talk about the things that we can compare with bacteria cells, or about the things that we kind of need to focus on to understand later on when we get to immunology. Okay, so just like bacteria, we said it's very common for bacteria to make some kind of glycocalyx, whether it's a true capsule or maybe just a slime layer. For eukaryotes, it's much less common. Okay, for eukaryotes, it's going to be anchored not to the cell wall, but to the cell membrane. Okay, so in other words, only cells without a cell wall are going to have a glycocalyx. All right, so that's some animal cells, some protozoa, not plants, not fungi. Okay, same kinds of functions of what we saw with bacteria, like attachment or protection from, not so much from phagocytosis, because it's the eukaryotic cells that would probably phagocytize the bacteria. But I said attachment. Definitely some protection against drying out. And it also does serve to strengthen the cell surface because there is no cell wall. So again, gives it a little bit strength, a little bit of protection maybe from osmotic changes. All right, so moving in, cell wall. This is hard to kind of sum up across all eukaryotes what the cell wall is like. So I'm going to do this kind of briefly. Remember which eukaryotes have a cell wall. Fungi, plants, algae. Okay, that's pretty much it. Protozoa do not have a cell wall. They have a protein pellicle surrounding them, and not a true cell wall. So, of course, not animals, right? We don't have a cell wall. The composition is really varied. A lot of fungi have their cell wall composed of polysaccharide chitin. There's some other molecules that are important there too. Plants, of course, cellulose is usually a major constituent, as well as algae. But there's a lot of variation within the algae. Some of them have like silicates, like the diatoms. Okay, but the important thing is composition never includes peptidoglycan. Okay, that is a strictly bacterial compound. All right, so you know, cell wall functions, strength, support, shape, same kinds of functions as for bacteria, prokaryotes. All right, so moving on, not going to spend a lot of time there. Again, the cell membrane, this should be kind of, you know, review. There are not a lot of differences between the prokaryotic cell wall and the eukaryote. That's why we don't really have, there's only a few antibiotics that target cell membranes because, right, they're going to be toxic to us as well. Of course, phospholipid bilayer, one difference is that eukaryotes have sterols for added support, or in other words, steroid lipids like cholesterol. And we all think, oh, cholesterol is bad, it's bad, but it's absolutely necessary to increase fluidity in cold temperatures and to stabilize the membranes in hot temperatures. Okay. So again, bacteria, no cholesterol. Function, of course, it's a semi-permeable barrier, like we said, for bacteria. Here's a unique function though, movement. Okay, if eukaryotes don't have a cell wall, then they can actually extend pseudopods. There's some cytoskeleton at work there that we didn't really talk about, but they can extend pseudopods to move in an amoeboid-like motion. Okay, some of our cells, some of our immune cells can move like that as well. Okay, we don't ever see that in cells with a cell wall because the cell wall is rigid, right? They can't extend pseudopods that way. Okay, so I'll just write amoeboid motion. Okay, and another unique function is transport by extending those pseudopods. Okay, so let's say endocytosis and exocytosis. Okay, there's two kinds of endocytosis depending on what the cell is trying to ingest. If it's ingesting solids, then we call that phagocytosis. And if it's ingesting liquids, lesser well-known name, penocytosis. Okay, and of course, exocytosis is when the cell is exporting substances, usually wastes. Okay, so endocytosing, bringing things into the cell, exocytosis, getting rid of them. All right, so I think these pictures are helpful there. Of course, this is a amoeba. It's extending a pseudopod here and a pseudopod here in order to ingest this unlucky paramecium. Okay, so it's endocytosing or phagocytosing by extending those pseudopods. Okay, again, a bacteria cell would not be able to do that because of that rigid cell wall. Okay. All right, moving in internal structures. eukaryotes that have flagella, they're considered an internal structure because they are membrane bound. Okay, most of the bacterial flagella, except for the axial filaments, are not membrane bound. So that's one difference right off the bat. I think I mentioned this before, but prokaryotic flagella and eukaryotic flagella, really the only thing that's the same is the function, that they are used to move the cell. Everything else is different. The composition, the energy that's used to do it, where that comes from, everything else is different. Okay, so eukaryotic flagella are composed of microtubules. Okay, and that comes from microtubules are composed of the protein tubulin. Remember, bacteria were made of flagellin protein. Okay, and that tubulin is arranged in a 9+2 arrangement. Okay, and that of course seems like a really weird way to express anything 9+2. But if you look here at this diagram, that just means the microtubules are arranged in 9 pairs around the outside and then 2 on the inside. So that's the 9+2 arrangement of the microtubules. There's also a basal body. I'll write that in here. So down here, for some reason, remember the basal body is where it's kind of anchored into the cell wall or the cell membrane. And for some reason, the structure of those microtubules change. They say a 9+0 arrangement. So basically it changes to 9 triplets and 0 microtubules in the center. And I don't know the significance of why that changes, but we do see that. So the eukaryotic flagella are just the filament and right hooked into the basal body. There's not any kind of that, hook region that we see for prokaryotic flagella. So the composition is different. The motion that we see is also different. And again, you can kind of get lost on YouTube looking for good animations of this. I found a couple that I think are very good. You can start with that. But the flagella motion for eukaryotes, it's described as being whip-like. So remember, our bacteria, they always spin around in a circle. We talked about when they spin one way, they go one direction. They spin the other way, they go the other direction. Or they might run and tumble, depending on the arrangement of flagella. Eukaryotes always move kind of in a whip. Okay, and I'll tell you the way that I remember the distinction there in a minute. This top video, let's see. This is a really long animation. It's like 11 minutes long. But especially at the beginning, there's some good clips of a protozoa. I think it's a euglena that looks kind of like this. And the interesting thing is that it moves in the direction of its flagella, which is kind of, I don't understand the physics of that, how that works, but it's kind of cool to watch. It moves so quickly that it's hard to tell that it's actually whipping. It almost looks like it's spinning around. But this second video compares bacteria versus a eukaryotic flagellated cell, sperm. And that's a little bit easier to see. I think that the sperm is moving in a whip-like, because I think it slows it down a little bit. The flagella are the bacteria are kind of buzzing around. But it's a good comparison, again, prokaryotic versus eukaryotic. Okay, and cilia, we're going to talk about that on the next slide. This is same composition as eukaryotic flagella, only eukaryotes have cilia. But this is how I can kind of remember that the eukaryotic flagella move in a whip-like way, because if all of these little cilia were spinning around, how would that get the cilia to like move in a directed way? Okay, they're all kind of beating in the same way. again, whip-like, and so that's getting that paramecium or whatever it is to move in a certain direction. All right, it's only eukaryotes that have cilia. Of course, these are really projections from away from the cell that are much shorter than flagella, stiffer, but again, same structure, that microtubule structure. Lots of them on the surface, maybe 100, maybe 1,000. What kind of cells have these? Protozoa have cilia, and really only a few types of animal cells. Okay. We have ciliated cells that line our respiratory tract. And that cilia, again, it beats in one way to help get particles that don't belong in our respiratory tract out again, like dust or maybe even bacteria. So again, it wouldn't make sense for those cilia to be spinning around in our respiratory tract. They're kind of beating in one direction. All right. So movement of the cell or just trying to circulate material. Okay, so then I'm just going to kind of buzz through this list of organelles pretty quickly. Again, not that it's not interesting, but you probably had some of this before in other classes. So just know kind of the basic function here. Obviously, the nucleus is where the DNA is kept. Endoplasmic reticulum is kind of a transport system. There's two kinds of ER. The rough ER has ribosomes. So remember, ribosomes is where the proteins are made, and it's going to make a certain kind of protein that's meant to be secreted outside of the cell in the rough ER. Okay, the free ribosomes are going to make all the proteins that are necessary for the cell to live. Maybe you have a cell that's making antibodies. Okay, that's on the rough ER is where it's going to make those antibodies. Those get secreted outside of the cell. The smooth ER doesn't have ribosomes. That's why we call it smooth. Has a lot of different functions like lipid synthesis, a lot of varied function. Okay, we're not going to get into that. Next comes the Golgi, where there's a lot of modifications being made. So maybe those proteins that came through the rough ER, maybe they get a sugar added to them, so they're glycoproteins, or a lipid added to them, so they're a lipoprotein. Okay, and it's kind of a membrane system to start to, transport things that are made maybe to other parts of the cell or out of the cell. The mitochondria is where ATP is made, powerhouse of the cell, and some eukaryotes may have chloroplasts, which is where the photosynthetic reactions take place. Okay, I want to spend a little more time on the lysosomes because later on in the course, we're talking about our white blood cells, how important they are to phagocytize invaders. The lysosome really gets center stage there. Okay, so the lysosomes are actually, they contain digestive enzymes that are made in the cell in the rough ER, transported through the smooth ER, and then kind of pinch off from the Golgi. So they're sacks of enzymes. And those lysosomes, their job is to digest any kind of invader that white blood cell would endocytize, like a bacterium. So the lysosome fuses with that membrane-bound sac that the cell is enclosed that bacterium in, releases those digestive enzymes, and most of the time is going to be successful in killing whatever invader that was. So again, especially important for phagocytic cells. We'll talk a little bit more about what kind of enzymes are in there later on, and the steps of phagocytosis later on. Okay, lastly, I want to talk, in chapter 3, we're making good progress here. We're going to talk a little bit about this endosymbiotic theory. Okay, so this involves the mitochondria and the chloroplasts as well, so we're going to talk about them in a little bit. But this is a theory that explains the origin of eukaryotic cells from some early prokaryotic cells. Okay, so we kind of assume that the simplest life on Earth were prokaryotic cells. And at some point, well, how did we get eukaryotic cells from that? Lynn Margolis put forward this theory in the 70s. And when she did and presented it at conferences, people would kind of laugh at her and get up and walk away. But today there's a lot of evidence for this. There's still some things that don't quite match up, but there's, and of course, it's not something that we can prove now, but there is a lot of support and a lot of evidence of this. So basically what this theory says is that there was some early anaerobic cell that ingested another cell. And instead of that first cell killing the second cell, they lived as endosymbionts. And that just means that one helped the other. Okay, it was a mutually beneficial relationship, and we had one kind of life living within another kind of life. So that's what an endosymbiont is. Okay, and that second cell that got ingested, that kind of became what we now know as mitochondria in chloroplasts. Okay, so what's some of the support for this theory? Some very interesting things. Both the mitochondria and chloroplasts, those organelles, have their own DNA. Okay, so that's kind of interesting. Lysosomes don't have their own DNA. Ribosomes, they have ribosomal RNA, but they don't have their own DNA. So that means that they can replicate independently of the rest of the cell. Okay, and another interesting thing is that both mitochondria and chloroplasts have their own ribosomes. And guess what? They're the kind of ribosomes that we see in prokaryotic cells. They are 70S ribosomes instead of the eukaryotic 80S. Okay, so the fact that they have their own DNA and they have their own bacterial ribosomes makes us think that they maybe perhaps were once free-living bacteria. Okay, and in addition, both mitochondria and chloroplasts are about the same size as bacteria. Okay, so that maybe is a little bit, well, that could be a coincidence, maybe, but again, there's good support that, you know, maybe that is how eukaryotic cells came about and where mitochondria and chloroplasts perhaps originated from. Okay, any questions before we move on? Okay, good. That gives us a little time to start chapter 12. Okay, so since we've just been talking about some eukaryotic structures, it makes sense. Let's spend a little bit time talking about eukaryotes. So the eukaryotes that we are interested in, of course, are the microscopic ones. So things like fungi, like yeast. Here's a protozoa over here. Both of these bottom pictures are algae. Okay, so we're not going to spend much time on algae, but maybe a little bit longer on the protozoa and the yeast, well, the fungi. Okay, so we want to first think about, there's a lot of variation among those groups, but, you know, what kind of constitutes an organism? Like why is it a fungi and why is it not a plant? That sort of thing. So we're going to talk about some kind of main characteristics of those taxa. I'm going to talk a little bit specifically about a protozoa because it's the one that Americans need to be most worried about when they're traveling abroad, and that's the protozoa that causes malaria. And then just a little smattering of fungal structures so we can kind of understand some of the terminology that goes there. This edition of the book cut out all its fungal reproduction stuff. And so I was happy to just cut out that as well. So a little bit less time on fungi maybe than what I have in the past. We do have a course that if you're really interested in fungal, it's diseases, fungal diseases that Dr. Cleary teaches. So if you're interested in that. So classification. So just to be straight on a few things, of course, we talked about how Linnaeus, he was kind of the first one to think of this hierarchy. He said plants and animals. Late 20th century, I talked about that Whitaker designation of different kingdoms. So the eukaryotes were fungi, protists, plants, and animals. The accepted taxonomy today is to have all these different kingdoms here that describe the eukaryotes. Are those in very much use? Not really, because nobody recognizes them. So we still kind of use the fungi, the protozoa to describe these things, but we need to keep in mind that it's really not that simple. Like some of these different kingdoms kind of, well, they're kind of like protozoa, but then they have some, you know, algal features too. So, you know, with reverting back to just saying protozoa, there's always going to be some oddballs that don't really fit into that protozoa group. Like this kingdom right here, Stromenopelia, those are parasites of plants. Just pull out an example here. Those organisms in that group were responsible for the great potato blight in Ireland. And those are described as algal protists that act like fungi. Okay, so just there are some organisms that really don't fit nicely into the designations up here that we try to squeeze them back into. All right, so let's talk a little bit about protozoa. What can we say generally about protozoa? Well, they're eukaryotes and they're unicellular. Most of them are motile. So we used to categorize them based on how they moved, whether it was with cilia or flagella or by amoeboid motion. Like I mentioned before, I'll write it again, they lack a cell wall. Some of them have this proteinaceous pellicle, okay, that kind of does some of the same things, but we don't call it a cell wall because it doesn't have sugars in it, it's protein. How did they get their nutrients? We describe them as chemoheterotrophic, most of them. Okay, so we're gonna pull apart that term probably next week, but basically that just means that they eat organic matter or ingest organic matter. Okay, troph means to eat. Chemoheterotroph just means they eat organics. Okay, some of them though, I'll put this in parentheses, some are photosynthetic. So they're actually considered photoautotrophs. So there are some variations within that group. Most of them are free living. You're gonna find them, we say ubiquitous in nature. That means they're everywhere, especially in bodies of water. But some of them are parasitic. And of course, by parasitic, that means they live at the expense of some other living person or animal. Okay, so here's some examples of some parasitic ones. Trypanosomes cause African sleeping sickness, among other things. Here's a water giardia, right? That'll give you a pretty good case of diarrhea if you drink water that's contaminated with those. These two, they look like eyes, but those are actually two nuclei, double nuclei there in the cell. Okay, and then of course, malaria is the one that I want to spend a little bit more time on. Just lastly here, just a little bit of terminology, very varied life cycles, but we can say that all protozoa exist in a trophozoite stage. All right, that's probably a new term. That just means a motile feeding stage. So in this graphic that I found here, this is a graphic from that protozoa that causes that, that you know, goes up your nose when you're swimming and causes that brain, it's Naegleria fowleri or something like that, that fatal brain infection. But anyway, that organism actually can have a couple of motile feeding stages. One's an amoeboid form and a flagellated form. So all protozoa have that. And then some of them can also exist as a cyst. And that means a hardy resting stage. Okay, so if the conditions aren't quite ideal, they can kind of hunker down into a cyst, kind of analogous to the endospore for the bacteria. This is not quite as hardy as an endospore, but it's going to enable the protozoa maybe to survive some conditions when they're not that great. Okay, so we'll start talking about malaria next time. I will post some more slides over the weekend. I hope you have a good weekend. I'll be in my office for a bit.